the one thing that you need to be aware of, learning these tools is fun, and studying these problems is fun, and digging into the data and things like that. But always the thing that you need to keep in the back of your head is, what am I doing this for, and how is this going to end up being applied? How does this eventually end up benefiting the bird, benefiting the producer, making our food safer,
right,
higher quality, inexpensive?
Good morning, everyone. Welcome to the Poultry Podcast Show. I'm your host today, Dr. Karen Grogan, and joining me today is Dr. Ryan Arsenault. Ryan is a associate professor in the Department of Animal and Food Sciences at the University of Delaware. He received a PhD in biochemistry from the University of Saskatchewan, where he conducted doctoral research at the Vaccine Infectious Disease Organization. And then he did postdoc training at USDA Agricultural Research Service. Welcome to the podcast, Ryan.
Thank you. Good to be here.
Excellent. So why don't you tell us about your current research there at Delaware, and what your interests are currently?
Sure. So my research here has kind of been a continuation of my postdoctoral work at ARS. I guess for those that don't know my background, my PhD work was in doing proteomics, and specifically cell signal transduction through phosphorylation, and studying host pathogen interaction with that technique, and brought that into the poultry world when I started at ARS, and specifically in gut health. And signal transduction, proteomics, that kind of omics work was relatively new in poultry disease pathogenesis. So we the initial disease was Salmonella, a food safety issue, an issue in the poultry industry that we're trying to grapple with continuously, it seems. And it's I think it's a particularly interesting issue because the chickens don't care.
Right.
For the most part, whether they have Salmonella or not.
No, the bugs are there and they don't necessarily get sick. They're just there hanging out, and then create a problem when it enters the food source. So
yeah, and I find that a fun problem to work on, because it's not a typical disease. It's not where you're trying to treat a disease, help the immune system to get healthier quicker. You're actually wanting the immune system to, hey, look at this. You should respond to this.
You should take care of this and make it go away. So what has your work led you to understand in terms of how is Salmonella sort of, is it invading the host cell, host immune cells? Is it hiding out? Is it not listening to signals? What has your research shown in terms of that?
So what we found is that if you give a chick, a newly hatched chick, a challenge with Salmonella, they will respond. They will treat it like a disease, and they'll respond to it in a way you would expect of other animals that treat Salmonella like an infectious disease. But if you wait a period of time, 4 to 7 days, it's about that fast, post-hatch, they will start to respond to it as if it was a commensal microbe in the gut. And we sort of tracked this. This was a lot of this work, the foundation of it was laid when I was, like I said, it was at ARS and then on to UD, where we tracked sort of this, we call it the tolerogenic response, where the immune system has this mechanism in place to tolerate the bugs of the microbiome.
Right.
And that's a good thing, and you want that because
we're getting those microbiomes are important, right?
But the chickens are doing that with Salmonella. Food safety concerning strains of Salmonella.
And what strains have you looked at that mechanism in? So what challenge organisms did you use for that?
So we've looked at Heidelberg, enteritis, and Typhimurium.
Okay. The three big important.
Right.
and so then, so if it's recognizing the Salmonella as a commensal, what strategies have you looked at sort of as a next step? So how can we alter that? Can we alter their immune recognition, or can we, try to decrease, increase the good gut flora to try to outnumber the Salmonella? That would be sort of a logical next step in your research.
Right. So it's interesting because the other disease that I've looked at sort of the pathogenesis of is necrotic enteritis. And
another complicated disease. You picked the simple ones.
Right. And there was sort of a deviation from the Salmonella work into the necrotic enteritis work while I was at UD, and now I've kind of like moved back towards Salmonella. So doing both at the same time has really made my perspective, I think, unique, or at least different than both the people that are strictly working on necrotic enteritis and the people strictly working on Salmonella, because the people strictly working on Salmonella often, the logical approach, and it is logical, is to induce some kind of immune response. And often the simplest one to attempt to induce would be an innate inflammatory response. But that is exactly the opposite of the thing you would want to do in the context of a coccidiosis or Clostridium perfringens challenge. So in the back of our minds is always, let's not make something else worse by attempting to deal with this pathogen. We certainly don't want to break more frequently with necrotic enteritis because we're trying to prevent Salmonella contamination of poultry products. So I'm leery of the attempts to sort of knock back homeostatic or anti-inflammatory responses to induce pro-inflammatory or pro-immune responses to the Salmonella. I think we have to be a bit more nuanced with that, because we'll cause problems in other realms of poultry gut health. So what we've simultaneously with all this work, because of the technology I use with the post-translational modification, we were actually able to look at metabolic signaling as well as immune signaling, and we started to integrate those. When I was doing my work designing the arrays to study the signaling, and these are chicken specific, so they had to be designed around the chicken genome and proteome. I had a metabolic array and a immune array that were designed separately. And for some reason, I decided I was going to put all the data into an interaction database and see if there were any links. And it turned out that they were completely cross-linked and there was no distinction between the two processes. And I thought this is really important, this is really interesting, and I thought I discovered something completely novel. And then I went to the literature and realized that on the human side, the concept of immunometabolism had been around for years. So we've really adopted that perspective when it comes to poultry health and poultry disease and poultry interventions.
Okay. So in your time with poultry, I'm sure that you realized, they're kind of on a knife's edge in terms of a metabolic plane.
Right.
So what I'm reading through here is we're doing ourselves no favors in terms of some of these, gut health conditions by keeping them there. We're in an industry that operates on fractions of cents. So everything is least cost. So if you're saying that, this is very, there's a metabolic factor here. What things are important to go into the diet of, the two things you're discussing are really important in terms of broiler health, in terms of necrotic enteritis, and then now Salmonella on the food safety side. So in terms of those two diseases in your work, what have you found that's nutritionally important?
So the other aspect of this, and a roundabout way to answer your question, is the other thing we've been looking at along with the post-pathogen interaction is a lot of the work, probably half of the work that I've done since I've been at University of Delaware has been studying the mechanism of action of antibiotic alternatives in the context of challenge.
Okay.
And we rarely see a good one that only impacts the immune system in one direction, either dampening it or activating it, and we rarely see that there's not a metabolic consequence as well. So the more effective ones seem to be what we call immunomodulatory. They sort of establish a homeostasis that provides a better baseline of response to a subsequent challenge. And that's across the board. We've looked at heat stress, cold stress, viruses, bacteria, parasites. And as you say, the metabolic aspect is part of it. Is we've likely made the birds, we've shifted them because of our production goals in one direction. And
Right.
the interventions, while they don't reduce productivity, they seem to shift them back to a more balanced state. Like you said, that's something that is always something we look at is we're already over here,
Right.
to start with. How do we get back to a baseline? And that's a better position to approach the
Right.
response.
I think that, I can appreciate, I think a lot of these products that we see coming onto the market as antibiotic alternatives are poorly understood. So I appreciate that you're doing that work to try to understand their mechanisms of action. And when you say immunomodulators, what has your research shown that they're actually doing? Like, what are you measuring in terms of what effect they're having?
So the sort of the spectrum of what you would expect if you were measuring immune and metabolic processes. So the major metabolic pathways: glycolysis, fatty acid oxidation, protein catabolism, and anabolism. Those are the general things we're looking at on the metabolic side. On the immune side, we're looking at the big innate immune signals, TLR signaling, the inflammasome signaling, and then the T and B cell. So we can sort of see, this is up, this is down. When you do this, these pathways are active or these pathways are turned off. And it depends on what challenge you're looking at and what alternative you're looking at to how the response breaks out. I've two students. One just graduated. She's actually at ARS now, Casey Johnson. She's looking at some botanicals.
Okay.
And she has a really interesting framework for how she sees what an effective natural antibiotic is. And I don't want to give away her approach because she's writing the paper right now. And then a student I have that's graduating this year from Auto Perry, she's looking at butyrate, and she's seen, again, this is an interesting, when you think about it in this sort of broader scope, we think of butyrate as either something that enhances efficiency or is anti-inflammatory or both. But she's seen in her data that there's actually a wide variety of gene expression changes, and it really acts as a immune altering, and not in the way you think. It actually will boost responses.
Right.
So it's not purely a feed component, and it's not purely an anti-inflammatory, because it'll get into the cell and it'll actually alter histone effects and change gene expression levels. So it's fun to pick this stuff apart.
Right. So I think that, the type of research that you do is sort of like the forefront, in terms of, proteomics and all of, the omics are kind of the areas where we're expanding our work. So you've looked at Salmonella, you've looked at necrotic enteritis. What's your next step in terms of, do you plan to continue with those challengers in terms of poultry health, or do you have new areas that you're expanding into?
I think those will be the ones we sort of stick with, because they're the biggest problems for necrotic enteritis in the production limiting sense, and for Salmonella in the regulatory sense. There's rumblings with FDA and USDA that Salmonella is going to get cracked down on harder. And I'm talking to producers, they're like, we will do what you ask us to do, but you have to tell us what to do.
What that is, what you want us.
Right. And will it work? So the producers are in a frustrating situation because they're being regulated, and increasingly so, but there's no tools for them to respond. So these two problems I am keen to stick with, and we're looking at feeding in effectiveness. I'm sort of agnostic about the alternative, what it will be, or if it's a macronutrient alteration. We just want to find something that works, and I am not a advocate of going backwards in terms of genetics, or going backwards in terms of our feed formulations to make a slower growing chicken, for example, or something like that. The poultry industry is too important in feeding the world to cut back on our ability to do that. I think advancing the science and advancing our understanding and solving the problems by going forward is the better approach.
Right. In terms of that, some of our opponents to the poultry industry say that we've created these things. So it's interesting in terms of, and a lot of the things they say that, we create Salmonella by housing, birds in the conditions in which we house them, that, chickens naturally, our opponents say that if we grew, just like you were saying, that if we grew chickens slower, we didn't grow them in intensive operations, that we wouldn't see the types of diseases that we have. Do you feel that those arguments are valid?
I think that there's overall no, because of what we do for limiting disease in poultry and biosecurity, things like that. So we are trying to prevent disease. We are trying, a perfect example is avian influenza, the outbreak that's going on right now. We're currently in the largest avian influenza outbreak in poultry in the history of the United States.
Yes, we are.
If the public knew the efforts that the industry and the producers and the state governments, et cetera, go to limit disease and to limit the spread of avian influenza, they would be amazed. I teach a first year lab class where we talk about biosecurity and all the different ways that we attempt to prevent disease. The food safety and the disease burden when you leave the barn, in my opinion, is much larger than when you're in the barn. The parasitic infections, the helminth infections, the predation, the stress they're put under.
I understand the stress is higher.
Right. So these ideas of like, we pack them together and thus this happens. There may be individual cases where a certain pathogen may spread more efficiently inside than outside, for example, but people are also often surprised when they go into a barn that they have room, they have space.
Right, space, and it's temperature controlled, and in the South when we get to 90 and 100 degrees and it's nice and cool and comfortable, and they are surprised when. So I do want to kind of circle back to something that you mentioned in terms of Salmonella. We are seeing increased regulatory pressure potentially coming from FSIS. And I think there is some, frustration within the industry of we are doing a lot of things and we have done a lot of things.
Right.
What is your research focus in terms of that, in terms of supporting? I know the industry there on the Delmarva is dealing with the same challenges. What do you see as far as, okay, maybe here's another tool that could help us in terms of controlling that food, those, the food safety concerns?
Yeah. And assuming that we can't find something we can't find the magic bullet in our research to induce the chicken to clear the Salmonella.
Right. You can find that, you'll be like, Nobel, prize winning. Right.
Exactly.
Yeah.
But I think it goes right from litter management, litter amendments, through to to the feed and the immunomodulation that we can apply to the processing plant. Like this is going to be this is one of those full spectrum approaches that is going to have
every tool.
Right.
I think that's part of what is not fresh. I think the industry realizes that we have to use every tool in the toolbox that we have in terms of salmonella control, and it starts, way up the line, like you were talking in terms of genetic selection and looking at the immune system, and then as it comes into the the broiler, using everything that you can in terms of like litter amendments, and but everybody wants the one silver bullet that will eliminate the problem. But I think like you were explaining that the bird is seeing it not as a threat, and it's seeing it as a commensal. So I think the challenge is even higher in terms of trying to get the bird to respond.
And the other aspect is that we've seen these shifts in strains. We've seen shifts from Typhimurium to Heidelberg to Kentucky
and Infantis.
And this is speculation on my part, but I wonder if our efforts aren't working. Right. It's just that there's this ecological niche that exists in poultry and in this system.
I totally agree.
And it just, the next one will.
The next one's going to move in. Right. Like we're okay. Now our focus is like, the focus was Heidelberg for a while. Okay, we got it down. We see Infantis come into play. And I think very much, there is a spot in the chicken gut and probably even other producing animals as well. There is a place for Salmonella, and that's where this bacteria has figured out, hey, I can just live a happy existence here.
Right.
But when it's regulated and the industry will have to respond and meet performance standards, that makes it a challenge.
Yes, and you're fighting nature at that point,
right?
I do think with necrotic enteritis, it's a similar problem, but we're not fighting the bird with it. So, for example, I do believe that there are these perturbations of the chicken gut homeostasis, whether it's a coccidiosis infection, non-starch polysaccharides, stress, that opens up a niche for CP.
The Clostridia, yeah. Absolutely. You just create, and I have customers ask me all the time, well, what is it? And I was like, it's this long list of things that could create this one little microenvironment that Clostridia is just going to bloom and grow. And I think that's what is, again, everybody wants an easy fix, and to be able to alter one thing to fix necrotic enteritis. And I'm sure your research has shown that it's not, like you're saying, it's those small what is could be one alteration can just be enough to flip the switch.
Right. And I think we probably have in the genetics and the feed formulation pushed the system for productivity purposes toward that tipping point. And without sacrificing the industry, how do we augment or ameliorate that? And then it's not unique to poultry. The dairy industry sees similar immunometabolic issues. We had I had a colleague here in Delaware that studied dairy cows and some of the hoof issues that they had, and he suspected, and I agree with him, that pushing the system, you're inevitably going to see consequences of that. But that's what we're for. That's what we're here for. That's what animal scientists, poultry scientists are here for, is to solve those problems.
So if you were a live production manager and you're dealing with necrotic enteritis in your complex, what's like the one message? People come in and try to sell you, like you were talking about, these antibiotic alternatives on the regular. What in your research, what are like I know it's a long list, but what are the say 4 key things that you found in your research in necrotic enteritis that our listeners could take out of this in terms of, here's some key areas you should focus on what they can do?
Yeah, for sure. The first one would be the environmental conditions. So keeping the litter as dry as possible, keeping the humidity controlled for any kind of growth that's happening in there. The coccidiosis is the obvious one that people think about, but the Clostridium perfringens, but the less stress on the birds and that the temperature, humidity, the litter quality, the litter, basically how wet it is, right? One, keep it and amend it and those things. And then another one's feed quality, which is hard for the producers to deal with, but maybe the integrators can look at that. Like sometimes, for example, I've heard people talk about sometimes the fats and the oils in the feed, if they're a little bit, if they're on the verge of going rancid, for example,
quality,
right?
Quality fats,
you're going to put that same, and that's a very clear stress that I could draw a direct line to the gut, because rancid fats, you can follow the secondary metabolites that are generated in the gut of those and how that inflames the epithelial lining. And CP, to me, CP is, it looks like it's a meat eater. So any kind of weird protein shift, including self-protein of the chickens,
right,
it's going to start eating it. And that's why we get those lesions and things like that. So avoiding that source of bloom for the CP is always going to be the thing in the back of your head. And stresses and all those panoply of stresses are the things to avoid.
Excellent. So Ryan, thanks for joining us in the Poultry Podcast. I think we've covered a lot of topics in our time today. What are what's some advice that you would give students that are, sort of interested in this sort of cutting edge technology that you're using in terms of proteomics or, the next phase research? How can they get interested in what you do?
The one thing that you need to be aware of, learning these tools is fun, and studying these problems is fun, and digging into the data and things like that. But always the thing that you need to keep in the back of your head is, what am I doing this for, and how is this going to end up being applied? How does this eventually end up benefiting the bird, benefiting the producer, making our food safer,
right,
higher quality, inexpensive? So I see that disconnect, and you see that sometimes with even in the poultry science world. There are people that'll look askance when you talk about, well, I'm going to present this omics work now, and it's like,
they're like, oh nope, I'm out.
Right.
Yeah. I'm not going to listen to this. I can understand. I can see that.
But it's our job as the people doing that type of research to say, no, it's not all just scary figures and spreadsheets and masses of data. It's
masses of data.
This is how it's going to. This is how we can apply it. This is how we can take it and benefit. And we need this approach as well as the classic approaches that we've been doing forever. Right. And it's the linking of those two, the feed trials, the large feed trials, and the things like that we need to see effect, like you said, because it's pennies. We're working with pennies per bird here, or less, fractions of pennies per bird. Yeah. And one of the easiest ways I explain this is, if we understand mechanism of action, that happens in every bird every time. When you do a large feed trial, depending on where you're doing it, the conditions, if it's a clean facility, a dirty facility, in the field, not in the field, a lot of things can alter that result. So it's hard to get a consistent fraction of a pound measurement every time. But a cell that interacts with a Salmonella, or a cell that interacts with a Clostridium perfringens or an antibiotic alternative compound, it's going to respond the same way, whether that bird's in Georgia or Delaware, a university facility, or a field trial somewhere.
Excellent. I think that the two things, overlap. I think we probably, in all these years of classic research, we've missed a lot of the things that you're now, looking at. And I think that all of the information, there's a wealth of information that's been out there. We just haven't been able to measure. So I find all of this work fascinating. And we really appreciate you being on the podcast today. And thank you, everyone, for joining in. And thank you for your time, Ryan.
Thank you. Glad to do it.